A continuación, les presento también con mucho gusto a la doctora Weina Sun. La doctora es viróloga y es profesora asistente del Departamento de Microbiología de la Escuela de Medicina ICANN de Mount Sinai. La doctora desarrolló vacunas universales contra la influenza y vacunas basadas en el virus de la enfermedad de Newcastle NDB para SARS-CoV-2 y otros virus. Estudia las respuestas de las células beta inducidas por vacunas y la inmunidad de mucosas con enfoques innovadores como la hemaglutinina en mosaico y la neuraminidasa modificada. Ha publicado más de 50 artículos científicos y tiene un índice H de 23, consolidándose como una figura clave en las estrategias avanzadas de vacunación. Agradecemos su presencia, doctora, y le invitamos a pasar al micrófono.
Seminar. I apologize I couldn't make it in person today, but my talk will be focusing on the data on the preclinical evaluation of NDV-based vaccine against COVID-19. As the previous speaker has already described, we built our NDV-based SARS-CoV-2 vaccine on the A virulent Lesotho strain of NDV, and we inserted the spike gene of SARS-CoV-2 between the PNM gene with important modifications, which is similar to what Dr. Paz has described previously. So basically, we code-optimized the nucleotide sequence of the spike protein for mammalian host expression. We then removed the polybasic cleavage site of the spike, and then we added hexaprotein prefusion stabilizing mutations. And last, we swapped the transmembrane domain and cytoplasmic tail of the spike protein with those from diffusion protein of NDV in order to increase the surface expression of the spike. And now we obtain a virion that abundantly expresses spike protein on the surface of the NDV, which we called NDV HXPS, also NDV hexapro-S vaccine. As you can see in this protein gel, as compared to the wild-type NDV virus, the NDV HXPS vaccine show abundant expression of the uncleaved S0 inside of the virion. Also, it has been described that NDV is avian paramyxovirus, so it grows really well in embryonated chicken eggs. And in the past several years, it has been well demonstrated by our partners, manufacturers, that they were able to produce both versions of the inactivated and live NDV-based SARS-CoV-2 vaccine in eggs with very little modification to their existing influenza virus vaccine manufacturer pipelines, and they were able to produce live vaccine and inactivated vaccine to be tested in clinical trials, either as a live nasal spray or intramuscular injection. So since 2020, we have been updating our list of NDV-based SARS-CoV-2 variants vaccine and up to KP2 as of now. I want to say all those variant spike protein inserted into the NDV adopt the essential modification that I just mentioned. But in other cases, we have to do a little bit more additional optimization of the antigen. For example, in the case of variants that has a P681R mutation, as shown here for Delta and JN1 variants, we had to revert the P681R back to the P residue in order to maintain the S0 contents to make the vaccine express a prefusion uncleaved spike protein. So with these variants vaccine in the past years, we have done work first to develop NDV-based mucosal COVID-19 vaccine for its ability to block infection and transmission, and two, to really formulate NDV-based variants vaccine into multivalency for its enhanced breadth of protection. So in this part one, I want to show some data on the mucosal immunity that the NDV-based vaccine can induce and how it can prevent infection and transmission. We all knew that intramuscular injection of many vaccine platform is highly effective against COVID-19, but it has very limited ability to induce mucosal immunity. So as compared to the natural infection, a live viral vector vaccine administered intranasally can more similarly induce mucosal immune response to block virus transmission. For the live NDV vector vaccine, we have validated this in our mouse model. As you can see here in mice that received 2 doses of either intranasal, showing red, or intramuscular live NDV hexapro vaccine with 4 weeks apart, they induce similar levels of serum antibodies, but only the intranasal vaccine were able to induce antigen-specific IgA in the nasal wash. And again, not only the intranasal NDV-based live vaccine can induce very high serum antibodies, as shown again here, the mice got 2 doses of intranasal NDV hexapro. It also has the ability to induce mucosal antibodies in different anatomic sites. So in this case, it can induce mucosal IgA in the nasal wash, also in the intestinal lavage samples, as well as vaginal lavage samples. So this data has the implication that NDV could be a very attractive vaccine vector, not only for respiratory tract viruses, but also for other pathogens that can potentially replicate or transmit through other mucosal routes. So then we wanted to test how well live NDV hexapro vaccine can prevent disease and transmission. So here we used the NDV hexapro XBB 1.5 variants vaccine and perform a hamster study where the XBB 1.5 vaccine was compared to the ancestral vaccine. And not only were comparing these 2 types of vaccine, we were also exploring 2 immunization versus 1 immunization. So these hamsters either got 2 intranasal vaccination with 4 weeks apart, or get 1 vaccination. And then we waited 6 weeks after booster and collect serum to evaluate serum antibody response. And as shown here, as you would expect it, that XBB 1.5 vaccine induce higher level of XBB 1.5 antigen-specific serum antibody response. And we don't really see a difference between the 2 doses and 1 dose of the XBB vaccine. But if you look at the neutralization titer on the right, it seems like a booster dose can slightly increase neutralizing antibodies. And what was also interesting is that we found the XBB 1.5 vaccine was able to induce neutralizing antibodies against more recent BA.2.86 lineage variants, including BA.2.86, JN1, and KP2 showing green, at least in the hamster models. And then for those vaccinated hamsters, we challenged them around 7 weeks after the booster to first evaluate protection. And then half of those vaccinated but challenged hamsters were co-housed with recipient naive hamsters 1 day after challenge for a direct contact transmission study. And for these animals, we collect lungs and nasotrabecinate tissues to look at virus replication for both donor hamsters and recipient hamsters. And then to evaluate virus shedding, we collect throat swabs on day 1, 3, and 5 to look at oral shedding of the virus in both donor hamsters and recipient naive hamsters. So as you can see that XBB 1.5 is not really virulent enough to cause any weight loss in the naive recipient hamsters. But if you look at on the left side, in the vaccinated hamsters, those that were vaccinated with 2 XBB 1.5 vaccines showing red were protect best from weight loss as compared to other groups. And then in the vaccinated challenge hamsters in the lungs at both day 2 and day 5, we do not detect any viral titers in animals that received the XBB 1.5 vaccine. And in the upper respiratory tract, we only see very low titers in animals that receive 1 dose of XBB 1.5 vaccine, but the virus was then cleared by day 5. So animals that receive XBB 1.5 vaccine were protected really well against the homologous virus challenge. On the other hand, in the naive recipient hamsters, we see no viral titer in animals that were co-housed with animals that received XBB 1.5 vaccine. So these data demonstrated that strain-matched variant vaccine can prevent virus replication better than the ancestral vaccine in the upper and lower respiratory tract and can prevent virus transmission. And we know that nasal cavity and oral cavity virus shedding is a very important factor for virus spread and transmission. So we look at viral load in throat swabs in hamsters, and we found in the vaccinated hamsters, those that received XBB 1.5 vaccine, the animals have much decreased viral load in the throat swabs. And we can tell that it looks like the 2 doses of vaccine reduce virus shedding much better than the 1 dose. So this is in the donor hamsters. When we look at the recipient hamsters throat swabs at both day 3 and day 5, so the recipient hamsters show in light blue here, it is very clear that recipient hamsters show a very low viral load in their throat swabs when they were co-housed with animals that received the XBB 1.5 vaccine. So this data demonstrated that even 1 vaccination of the matched vaccine can protect really well against virus transmission in the hamster model. To summarize the part 1, I've shown that intranasal vaccination of the live NDV-based vaccine can induce really strong serum antibodies as well as mucosal IgA response in mice. And I have shown that intranasal vaccination of the live NDV-based variant vaccine can prevent viral replication and direct contact transmission in a hamster model. So in part 2, I want to talk a little bit about our data on formulating NDV-based multivalent vaccine for enhanced breadth of protection. So this is based on our previous work where we found that intramuscular vaccination of a inactivated trivalent vaccine, which include ancestral beta and delta variants, can actually confer protective immune response against an unmatched Omicron variants to show a cross-neutralizing activity. So what we did afterwards is we want to continue to investigate this multivalent formulation to look at its immunogenicity and protection in a hamster model, but in the context of live trivalent vaccine. So here we test a live trivalent vaccine either given intranasally or intramuscularly. I want to mention that each component has 1/3 dose of the total. And of course, these trivalent vaccine will compare with their monovalent ancestral vaccine counterparts, either given intranasally or intramuscularly. So these hamsters were vaccinated twice and then challenged with an unmatched BA.1 Omicron variants. And when you look at the serum antibodies, it is very clear that trivalent vaccine showing dark pink, either given intranasally or intramuscularly, they induce very similar neutralizing antibodies against the ancestral virus, and the level of neutralizing antibodies is comparable with their monovalent vaccine. But in contrast, when we look at neutralizing antibodies against the BA.1, we found that both versions of trivalent vaccine induce higher levels of neutralizing antibodies against the BA.1 than their monovalent counterparts. And more importantly, we found the trivalent intranasal vaccine induce higher neutralizing antibodies against the BA.1. So this is very similar to what we saw before with the inactivated vaccine. When we look at the virus replication in the tissues of those hamsters, again, we saw that both trivalent vaccines showing dark pink, they reduce virus titer to the lowest level at 5 days post-infection. And both versions of trivalent vaccine reduce more virus titer as compared to their monovalent counterparts. And in lungs, we found that by day 5, all animals cleared their viruses in the lungs. And in terms of virus shedding from the throat swabs, again, we saw the same trend where the trivalent vaccine given intranasally showing discontinued line or given intramuscularly showing solid line, they reduce virus shedding much better than their monovalent counterparts. And again, the mucosal trivalent intranasal vaccine reduced virus shedding with best efficacy. So basically, these work led us to really do more formulation, hopefully to include recent variants such as JN1 and KP2. So first we did is we looked at immunogenicity of the monovalent JN1 and KP2 vaccine in mice. In this case, the JN1 and KP2 vaccine was compared to either XBB 1.5 or ancestral monovalent vaccine. And these vaccines were either given intranasally or intramuscularly as a live vector vaccine. Animals received 2 doses of the vaccine, and we look at serum antibody response 2 weeks after the second dose. As you can see on the right, that all viruses induce neutralizing antibodies to the highest titer to their homologous virus. And importantly, we saw that JN1 and KP2 vaccine induce similar levels of neutralizing antibodies, and they neutralize each other pretty well. In terms of mucosal antibodies, we found that, again, JN1 and KP2 vaccine given intranasally induce similar levels of mucosal IgA in the nasal wash against the KP2 RBD. So these data demonstrate a very good immunogenicity of the live intranasal JN1 or KP2 vaccine. So the last data I want to show that is we actually done some work to generate new multivalent formulations, including the most recent variants. In this case, it's the KP2 variants. And here we have the 2 trivalent formulations. The first one is BQ 1.1 plus XBB and KP2. And the second one is Delta plus XBB 1.5 and KP2. And as you can see that while BQ 1.1 XBB plus KP2 broadly neutralize most of the Omicron variants, Delta plus XBB 1.5 and KP2 actually can broadly neutralize both historical and recent variants and almost all the variants tested in this panel. So these data combined really show the great promise of multivalent NEV-based vaccine for enhanced breadth of protection. To summarize part 2, I have shown that the multivalent formulation of the live NEV-based vaccine administered intranasally or intramuscularly can induce better protection against the Omicron variant vaccine in hamsters than the monovalent vaccine. I have also shown some data that multivalent formulation of the mucosal live NEV-based vaccine consisting of the most recent variants could have great promise to offer broad protection. So collectively, we believe NEV-based vaccine platform is really good vaccine platform because it's cost effective. And we have seen from clinical data that both live and inactivated NEV-based vaccine are safe in humans. And the live NEV-based vaccine can induce both systemic and mucosal immunity. And additionally, NEV-based vaccine could be formulated as multivalent vaccine for broad protection. Last but not least, previous speaker also showed that NEV-based vaccine platform can be adapted for many other pathogens to show its universality, such as NDV-H5, like Adolfo mentioned, and also NDV expressing fusion protein of human RSV or even bovine RSV, which are also in development. This work is a huge collaborative effort from my lab and Peter, Adolfo's lab and Florian's lab, and I want to not acknowledge our manufacturer partners, especially AVMax, and our funding sources. And thank you so much for your attention.